network of classical conditioning on which we built everything that followed. But so Ton is a theoretical physicist, and in this whole neural network revolution of the 80s and 90s, Ton really came at that from a theoretical physics perspective with the idea to sort of give rigor to the field and place it on a more formally defined framework where he made really important inroads. Then he moved to King's College in London, where he has been now for almost 25 years, or 20 years?
Since 1995.
Well, you do the math. You're the smart guy.
I'm not good. Only with Greek symbols, not with numbers.
Okay, good. So, for a long time. But then he moved really from the field of neural networks to molecular biology, signaling pathways in cellular systems with a particular focus on cancer. And in doing that, Ton has touched upon many issues, going from information theory to network analysis and anything else you can think of, just to strengthen the formal base of our understanding of these networks. These are very generic tools that are also of great relevance for the study of the brain and other kinds of biological systems. So it's really great that we have Ton here. He's also translating this work now to a spin-off company where they're trying to have an impact on how we deal with big data, which is another really exciting adventure. So it's fantastic that Ton is here. Thank you very much for joining us. Welcome to BCBT, and looking forward to your talk on neural networks versus immune networks.
Thank you, Paul. And yes, these were great days when we met first, because in those days it was perfectly normal that a PhD student in a psychology department would team up with a theoretical physicist in another university and just get on with it and enjoy themselves. And I think it was a case of us having similar levels of ambition and similar views on the necessity of supervisors. I think that's sort of the common theme. Okay, now this talk is about the immune system, and the clicker works. Perfect. And the relationship... Oh, I have to stand here anyway. And the relationship...
No, if you want to walk around.
No, I can stand here. And the relationship between models of the immune system and models of neural networks. And this is a very interesting, and it has been a really fruitful interaction between these fields. But first I need to tell you a bit more about the immune system so that you can see for yourself what this similarity is. I'll start with description of the main players, the main components, the task of the immune system, why this involves learning and memory. Then the motivation, why am I at all bothering to study the immune system? Why am I so interested in it? And then focus on that similarity. And this similarity was picked up long ago. But very recently it became clear that it now goes much more beyond similarity, almost to, at some level, mathematical equivalence. Then, after having done that, I want to step back a bit and ask about what have we achieved in trying to do mathematical models of recurrent neural networks, because we need to understand how the boundary of the technology has moved forward and why certain things were not possible in 1995, let's say, but are possible now. And it is of relevance to the immune system. And then move on to actually show you some really interesting recent work that was done by a group at La Sapienza in Rome. And these people roped me in because at some point they ran into technical problems that were very similar to things that I'd done in the past. And I was very grateful for that because it was really interesting work. And show you some further developments, what is going to happen next with these models, followed by discussion and the usual acknowledgement. And I'll list some of the relevant references at the end. So I'm not an immunologist, I'm not a neuroscientist. I enjoy solving models, but specifically models that have some relevance to the real world. And that's why I went into this area. Okay, let's start with immunology. It's a fascinating area. But the way I see it is that the immune system is an extremely powerful system. It can kill you within hours if it wants to. So we all have this nuclear missile in our basement, and we protect ourselves against diseases using sticks and stones, and we have this marvelous technology, but we cannot control it yet. Now, things are beginning to change, but that's why I'm so interested in it, because I had for a couple of years I've been working a lot on cancer data, and then you see this and you think, this is remarkable technology. This is the future of medicine, not just in cancer, anywhere. So I find this really interesting. So let's start looking at what it is. what it looks like. So the role of the immune system should be clear. It should protect us from invaders, and they can be invaders from the outside, like bacteria and viruses, but it also protects us to some extent from degenerated cells that we have inside ourselves and were harmless but became dangerous, like cancer. And the immune system has developed, of course. There's one part of the immune system which is, in terms of evolution, really old. It's the innate immune system. And that's a very generic protection mechanism. And it works within ours. It is in all plants and animals. And what it basically does, and I'm simplifying everything because this is an enormous field in itself, but in a nutshell, what it does, it recruits immune cells to the infection site, and it does that via cytokines, small molecules that are released and that have a chemotactic effect. Then it creates all kinds of physical and chemical barriers for bacteria. It also sensitizes pain receptors. And it activates, let's say, the big guys, the adaptive immune system, which only happens in vertebrates, but not in plants, for instance. And all these things make us experience what we call inflammation, and this happens within hours. So the cells involved in this innate immune system, they are not specific to pathogens. They sort of look for generic things that shouldn't be there. And there are various types of cells, and the most famous one probably is the natural killer cell, which just goes for anything that shouldn't be there and tries to kill it, specifically cells infected with viruses. But you have other cells like mast cells. Macrophages are like the hoovers of the system. They look for anything that is not meant to be there and just eat it and digest it, and that's the way to get rid of it. And other cells that have specific roles to play. And immunology, as we will see, a lot of it is still unclear. It's much less advanced in terms of what we know about the hardware than neuroscience is, for instance. And then come the second wave, and this is happening within days. This is called the adaptive immune system. This is a more sophisticated response. And it also developed later in evolution. Plants don't have it, only vertebrates have it. And just to list, first of all, what is happening, and then we can ask how it is doing all these things. It develops a response that, unlike the innate immune system, is highly pathogen specific. So it's not a response to anything. It's really tuned to what actually are the characteristics of the invader. And it involves learning and memory mechanisms. It can memorize an invasion and the characteristics of that invader. And the way it does that is by tuning receptors. And in these cartoons here, you see that there are very sophisticated receptors on the outside of the immune cells. And the key thing of the adaptive immune system, which distinguishes it from the innate one, is that these receptor properties can be modified via genetic mutation and selection mechanisms. So if a certain receptor has some way of connecting to an invader, and this invader is a dangerous thing, it will itself adapt the adapter to the next time recognize that invader even more effectively. So this is fairly specific and fairly sophisticated. It also involves all kinds of complicated cell-cell communication, and that's where you can already feel a hint of neural networks coming in, and that will indeed happen. And all of this results in an enhanced secondary response. The next time this same bacterium or virus or whatever it is comes in, the system has memorized the features, and it has already tuned the receptors of its defense mechanism to recognize specifically those things. That's why the next invasion is dealt with much more effectively, and you might not even notice that you've been infected again. And that's the basis also of inoculations. So these cells that are responsible here, they have very intelligently adapting pathogen-specific receptors. And there are basically two classes of cells. It's not that clear-cut, but roughly in order of magnitude, leading order of magnitude. You have B cells, and the B refers to the fact that they originate from bone marrow. And we have T cells that are born in the thymus. And they coordinate their activity. So let's see how that happens. So what is the strategy? What is the, if you want, the thinking of the adaptive immune system? It's in a way fairly simple to understand. What it basically does is it makes sure that B cells can recognize specific antigen types and put a sticker on it whenever they see it. And that sticker is in the form of what we call antibodies. Yeah? So B cells release antibodies whenever they are activated by an antigen. Antigen is the name for something that comes and that can be detected. And then just like I illustrate here, you just mark it. You say this thing needs to be destroyed. And then the innate immune system has all these players that just go around, and anything that has a sticker on it, an antibody on it, out it goes. It's being removed, it's being killed. So this is, in a way, the principle of the adaptive immune system. But this process, of course, needs to be controlled really, really carefully, because you don't want those stickers to go to the wrong place, right? You have to do this extremely carefully. And what has developed is this intricate play of B cells and T cells, where the B cells are the cells that do the job of producing antibodies, and the T cells are like managers. They're controllers. Yeah? And the B cell will only be able to be activated and release antibodies if it not only detects antigen, but it also gets a control signal from very specific T cells that have also detected the antigen and produce bits of it to the B cell, and have to be all kinds of complicated chemical handshakes here. Yeah? So the B cell, there are B cells that can go for it even without interaction with T helpers, but that's a small fraction. Most of them need very complicated go signals from the Ts. And only then will they be allowed to do what they do. And then later it was found out that there are in fact two types of T, and it's just like excitatory and inhibitory neurons. You have those that can activate B cells, but you also have what they call regulator T cells that can deactivate the B cells. And the T cells also require antigen. So it's a very complicated thing involving handshakes and very specific interactions. So these Ts are not interacting with every B, but only with very specific ones.
So
yeah.
You say these control signals are complex, and I definitely believe you, but what's the space of complexity? What's the magnitude of complexity that I have to sort of think of?
Well,
about thousands, millions, billions?
Well, I'm not thinking complexity of numbers, but there has to be a really very specific matching of receptors that, so you get, it's like docking. You have Ts that can dock to specific receptors of Bs, and they will choose only very specific partners to dock to. But all these things are plastic. They can change. Yeah? And that's why it's an adaptive, tricky system.
So this lock system, how big is that language? How many elements do we have? Like ligands and receptors. What's the order of magnitude?
I think it involves quite a couple of genes that are modified in these adaptable parts of these receptors. But I don't know the numbers out of the top of my head. There are papers on specifically counting those things, because a lot of that was done in the 90s when they looked at shape space, and they thought in terms of barcodes, and they were thinking, how many different barcodes can I make in order to do these things? So there are papers on that. So, and these cells, it's not as simple as saying we have Bs and Ts and that's it. You know, as in biology, the more you start digging, the more variability you see. And there are all kinds of roots that tell you how these things are coming into life. So you start from stem cells, and then you get specification, and you get T cells, B cells. But even within the B cells, you have different stages. You have naive B cell, that's a B cell that hasn't seen antigen yet. It has different properties from a B cell that has seen antigen, which is called a mature one. And then you move further, they become later plasma cells, which means that they've been activated, and now they start specializing into producing enormous amounts of antibodies when they are plasma cells. Similarly, Ts, you have naive Ts that haven't seen antigen yet, and you have activated or armed effector Ts that have seen the antigen. And even then, I mean, if you look into the bone marrow, people have all kinds of classifications of stages of B cell, but if you really look at the data, it's a bit dodgy in the sense that certain classifications are somewhat ad hoc. If you use a different clustering algorithm, you get different classes of B cells. But a lot is happening in the lifetime of a B cell, and that's why I'm showing this picture. So they are produced, you get a B cell, it encounters antigen, at some point it goes into what is called the germinal center, where the B cell is brought into contact with all kinds of other things, and I'll come back to that, in order to make sure that you don't get B cells that attack your own cells. But a lot of things happen, and you cannot model all of that, so you have to simplify at some point. But the greatest problem that the immune system, the adaptive immune system has is this. It's called the self-non-self problem. How do you make sure that the immune system doesn't start attacking your own cells, right? Because if you just generate B cells with random adapters on the outside, random signaling molecules, they will at some point also respond to a cell or to something produced by a cell that is your own cell. So you don't want these crosses to go on your own cell. And of course, sometimes that actually happens. And you can get false positives, which means you get autoimmune diseases, or false negatives, where you get infections that aren't leading to a response. And what's even more tricky is if you have cells that are self cells, should not be attacked, but in fact, they become cancerous and you want to attack them. And in fact, the immune system is remarkably effective. This is, you see T cells, cytotoxic T cells, attacking a cancer cell. And some of these films, you can look at all these films on the internet, is really very encouraging. It can do enormously powerful things, but it doesn't always do it, the immune system, and that's where the science comes in. But this is a big problem, the self-non-self problem. Now, learning and memory. Why do we say that there must be learning and memory, even before we could find where it happens? First of all, resource limitations. There are so many shapes that can come into your body. You cannot maintain a B cell population in which all these shapes are represented and ready to act. It's just not possible. The second reason is if you can learn about shapes, you can improve the efficacy of your antibody binding. If you know what things to look out for, you can adapt. And the other thing, before you are released into the wild as a B cell, you need to learn who is your friend and who is your enemy. And that's what indeed happens. And the mechanism is this. It's called hypersomatic mutation. At some point when B cells or T cells are responding, are noting something on their receptors, they get through a process of hypersomatic mutation. Mutation rate is all of a sudden cranked up in these so-called variable parts of these receptors. And there is a selection mechanism where those B and T cells that respond significantly to self molecules, self-antigen, they get killed. And also B cells that are never triggered, they get killed because you're just wasting resources. So they're thrown into this pool, into the specific area of the body where they undergo this mutation, cranked up mutation. And then you kill all those that never respond, and you kill all those that are permanently activated, because the assumption is they're responding to self. So that's where it happens. And then a bit of terminology. We have the concept of clones. A clone is just a group of B cells that have the same receptor, basically, a group of T cells that have the same receptor. So they respond to the same enemy. And this is what happens now. Next, we want to ask, okay, so if we can adapt these receptors and make them more effective, and then at some point we've released these antibodies and the innate immune system has removed the enemy, now we want to remember. We want to memorize what particular antibodies were so effective. How is that done? Now, at the moment, consensus is, but I find it a bit of a cheap way out, is, okay, you know what we assume, and there is some evidence for that, of course, that after the immune response, that the B cells that were really doing the business, that were activated, that were responsible for removing the antigen, they become very long-lived cells. Yeah, they change into a shape where they can live for 10, 20, 30 years. It is one particular explanation, but there are others, and some were very popular some time ago. This is another one. Jern's idiotypic networks. He launched this idea in 1974, and he got a Nobel Prize for it in 1984. But now many biologists think we shouldn't have given him the Nobel Prize because it was rubbish. They were a bit quick, and they no longer believe this is true. But what's the idea? Well, it was a clever idea and a very natural idea. What Jern basically said is, if you have some activated B clone, it releases antibodies. Chances are there will also be other B cells who are responding to those antibodies who have, let's say, opposite structural features in their receptors. So you will, let's say, if you have a clone activated, you will also start activating the anti-clone, and you will get anti-antibodies. And before you know it, you have a state, and often you see this picture, because it's not one-to-one. Yeah? Antibodies can react to parts of an antigen or parts of another antibody. So you get all these combinations of different B clones keeping each other in an activated state via this mechanism of antibody, anti-antibody. So the system can be in two states. Everything is quiet or everything is activated. And what Jörn proposed is that you remember past infections because the antibodies and the anti-antibodies of those keep activating each other. So you have these combinations of activated clones. But as I said, nowadays, biologists have been moving away from this idea a lot, although there are some people, and this continues, who are developing this idea further. Because at the time when he did this work, many of the lymphocyte types weren't even known that we now know are there. So there is room for improving this idea. Okay, so and this fact that many, yeah?
Because that's a bit the idea of a reverberating circuit, right?
Yeah.
It's like a heavy in memory.
Yeah, you keep in. So you have a flip-flop, bimodal state. You can all be together. Everyone's happy and quiet, or everyone's happy and shouting all the time. Okay, so as I mentioned a couple of times, immunology, and we should not forget that, is a much more undeveloped science compared to neuroscience. And if you just look at some of the years where things were being discovered, only very recently, I would call this recently, 1995, that discovery of regulatory Tcells, these inhibitory Tcells. This is not so long ago, right? And if you look at the canonical textbook, I mean, I got recommended this one, so the Janeway book. Nearly every year there's a new edition, and nearly every year it has 20 extra pages, and there's all kinds of things that have changed in these books. That's how this field is moving forward, immunology. So there are many things people believe now that in 3 years from now they might say, no, it's not like that, it's more complicated, or there are other players that we didn't know about. So this is an evolving field, pretty much evolving field. Now, why am I interested in this? As I said, I've just been enormously impressed by the power of the immune system. So let's look at that a bit, and this is some work that we did in a completely different context. It tells you something about the prognostic power of immunological signals. And I need to first explain a bit what these curves are. So these are curves that happen to be, they were computed for cancer data, breast cancer data. And what we did is we had data on people in the form of measurements and time to event. And time to event in this case was disease-free survival. And then you have particular regression models where you try to predict basically the time where these patients get a disease relapse. And what you can then do is you can run via cross-validation, you can run analyses on all the variables you have, all the covariates, and you do the prediction. Normally you will get overfitting, then you throw out the least relevant variable, you do it again, and at some point you have a mechanism for defining the optimal variables with which to predict time to event. So that's all happening behind the scenes. That's not what these curves tell you yet. But we have this fairly standard. And then what you can do, you use the optimal variables that give you the most prognostic information about the progression of the disease, and you plot these curves. And these curves tell you about, let me see, about the performance, the fraction that you can predict correctly with your regression model as a function, the fraction of outcomes you can predict correctly. So you try to predict here, for instance, after 2 years, for all the patients in your data set, how many do I expect to have seen a relapse in their disease? And then you count, how often did you get it wrong with your regression model? And then the black curves, the black dots is the performance measured on the unseen data. So you always set aside some data that were not used, and the whites are the training data. So the black ones are the important one. Dashed line is just the baseline. What could you have predicted purely on the imbalance of the patient without looking at any markers? So the way to read these things is this. So this is the number of years. If you see a gap between the dashed line, the baseline, and these black markers, it means that you have been able to predict outcome for these women significantly better. Yeah? The fraction that you can predict using the information is much higher than what you could predict if you didn't use the information at all. Now, and then you see something really interesting. If you compare the standard things that medics have been using all the time, like tumor size, grade, number of lymph nodes involved, things like that, the standard variables, and you compare this to what you can predict in terms of outcome by looking at immunological markers. And these are densities, location parameters of lymphocytes, even in lymph nodes where the pathologist says, oh, there is no cancer in here, it's irrelevant. Don't even look at it. No, look at it. Measure what the lymphocytes do. Then you find that if you do this for all breast cancer types, not only do you find that there is information in these immune markers, you are above the dashed line, but if you combine the two, you get better prediction. You get many more cases right, statistically significantly. And this is even stronger in triple-negative breast cancer. Triple-negative is the one where you don't have estrogen receptors, you have no progesterone receptors, you have no, what is the other, where Herceptin works. So there are 3 hormone receptors and they're all absent. And what you see here, if you try to predict clinical outcome for these women, these standard markers tell you nothing at all, completely pointless. Your clinical outcome is completely independent whether you had a big tumor, high grade, low grade. But if you look at the immune markers, then you can start predicting things. And if you combine them, it's even more powerful. So this tells you that the state of the immune system is an increasingly interesting and powerful predictor for what will happen in disease. Another example, many of you might or might not have heard of this particular trial. This was a medical trial that was carried out in the UK at some point that involved a genetically engineered antibody that can activate T cells without needing any antigen receptor. So you take away the need for there to be anything. And this was a dramatic mistake. So it was a small-scale trial. It was an early trial. 6 volunteers were given the drug. Within the hour, they were all seriously ill. Within less than a day, they were all in intensive care. And they were only barely kept alive over months. And what they experienced was cytokine storm, where all of a sudden there's an enormous release of the immune system really completely explodes into a complete uncontrollable state. They had multiple organ failures. Some even lost fingers. I mean, this is a real picture from people that were in this trial. Long-term effects, they've never recovered their immune system and they will suffer from all kinds of immune-related problems for the rest of their lives. And this is what I meant earlier when I said the immune system can kill you in hours if it wants to. You just hope and trust that it doesn't do that. And looking back, there were a couple of mistakes made in this trial, procedural mistakes. First of all, they extrapolated what they call the safe dose from animal studies. But these genetically engineered cells were engineered on the basis of human data. So if it doesn't really have a very strong effect in the mouse, doesn't mean that it won't have a strong effect in humans. And the other mistake they made, they gave it to all volunteers at the same time. They should have given it to one and then waited for a day, and then they wouldn't have given it to all the others. But the interesting thing for me is that no one still knows what actually happened. What was the mechanics of this disaster? So it's an amazing, powerful system, but we don't really understand it in full. Now, things have moved on, and what is a really interesting development in cancer immunotherapy is this now. You want to not mess around with the antibodies, with the cells directly. And one of the more recent successful stories is what is called CART, chimeric antigen receptor T cell. So what they do is they take T cells out of a patient's body, and then they genetically modify the T cells on the basis of tumor properties of the same patient. They create then very high affinity antigen receptors in those T cells that can respond very strongly to the tumor of that patient. And in the next generation, they also have extra co-stimulatory signals. And these have been very successful in leukemia and lymphoma types, cancer types, not in solid cancers yet. But the interesting thing here is sometimes when there are problems, it is because it works too well. Now that is a situation that we normally have never seen in this field. It works too well in the sense that these are cells, so they multiply. And there is really no correlation between effect and the dose that you give, because you can just give a small number. They start multiplying. And what some of the problems related to was, again, the creation of these cytokine storms, uncontrolled proliferation of all kinds of other lymphocytes. And this, which is really a very strange thing that we've never seen before, it kills the patients because the tumor is destroyed too quickly. And what happens is all this tumor, dead tumor material, is released into the body. And the kidneys can't cope. The kidneys shut down. So in a way, the problem here is it works too well. And the problem is how to control this, how to make sure that we have some way of handling the attack that is going on. But what is really nice is that not only has it been proposed to be licensed recently, in fact, yesterday, this was yesterday late afternoon, FDA has now approved this. So this is now state of the art. It is beyond experiment. It's now actually going to be approved for patients. So this is really exciting. Now let's look at the similarity. Why do we think that there is a similarity? And then move on to what we can learn from it with neural networks. Well, first of all, both systems are recurrent systems with many variables and with parallel dynamics. That's what they have in common. It involves adaptive interactions between components. That's what they have in common. And distributed storage and processing of information. And then you can make a full table and look in a bit more detail, compare immune networks to neural networks and computers. So we have in all cases large numbers of components, although in the case of neurons it's more than in the immunology. Here the variables are concentrations of clones. Here there would be spike trains. The immune system is also slower. It works in hours, days. Dynamics is parallel. The links between the components are adaptive. But the two differences are here at the bottom. So the red. In neural networks, most neurons talk to a very large number of other neurons. We're talking thousands at a minimum. Yeah? In the immune system, it's low connectivity. So we're talking about maybe 5 to 10. And the other difference is that we know the equations, maybe not in 100% detail, but we know Hodgkin-Huxley equations. We have all kinds of simplified versions of it. But here, we don't even know what equations to write down. We can write phenomenological things, but we're not at the level where we actually have this detailed set of equations. So that makes the modeling harder. But the thinking in why do we think we can learn more from neural networks is basically this. The nice thing of neural networks is that we know how to program them. And I'm just writing the most primitive things here, and there are other ways in which we can reprogram neural systems. But if you just think about Hebbian type rules, in the language where you choose, let's say, plus or minus 1 for your neural variables, if you update your synapses in this particular way, then you're basically imprinting an instruction to the system. You're saying that if you find yourself in this state, you go to that state. And we can also deprogram them. We can take this type of instruction out, just with a minus sign. If we reduce the synapses collectively according to a recipe, then what you're saying is if you find yourself in state psi, don't go to state eta. And this is nice because it allows us to manipulate the landscape in neural networks. I mean, in practice, it's another thing, but in principle, we can learn, we can unlearn, we can control the response to triggers. Now, all of that we don't have in the immune system, but imagine if we would find something like that, what could happen? So the first is, because the systems are similar, well, this is clear that we can use intuition, experience, and this is already happening. But what if we could actually reprogram the immune system? If we found something equivalent to the HEP rule, then I could just tell the immune system, look, if you think about things like thyroid cancer, breast cancer, organs that you can live without. Yeah, you can have medication, you can take your thyroid out. If you could just tell the immune system, from now on, thyroid is your enemy, it would change everything, right? We would have a completely new ballgame in medicine. And the nice thing is we know the immune system can do it because, you know, there are diseases like alopecia where from one day to the next your hair fall out. It's a switch just like that. It doesn't take years. One day you're fine, next day you're bald, right? It happens. So, and that's when the immune system decided a certain cell type is no longer self, but it is non-self. So imagine that we could control this process. But to do that, we need theory. And we need models. So let's now look at how have we done it for neural networks and where are the technical problems and what is possible and what is not possible. So I want to step back a bit to The idea is behind statistical mechanics. So statistical mechanics is just a way of approaching problems with many variables, and physicists have had to do this because they wanted to understand simple things like why is water freezing, right? Why do magnets exist, and why if you heat them up they're no longer magnetic. And they've learned pretty quickly that yes, you might know all the equations that govern the evolution of all your particles, but there's no point trying to solve these because you have 10 to the power 24 equations. Forget about it. And instead, the strategy has become this. You take all these equations and then you derive new equations for quantities that are more macroscopic in nature. And those you solve. Yeah? So the trick is in going from this level of description in a clean way to this level of description. And then you get all kinds of things happening. Yeah? If you look at, and the nice thing is it only works for large systems, but in reality, if you do simulations with whatever system you like, large already means 1,000. A system with 1,000 interacting particles behaves more like an infinite one than like a small one. So you find things like phase transitions, and you find this nice properties called self-averaging, that in these large systems, you don't need to know the values of all your microscopic parameters because the macroscopic behavior will only depend on statistical properties. Yeah? If I look at a liquid of water or some other molecule that might be completely different, they all freeze just at a different temperature, but they all generically have the same behavior because details become less important. So this has been done, of course, for neural networks, and that was at the time when I did my PhD. This was very popular, and a lot of work was done. Because you can follow this strategy, and it works, right? You have large numbers, tick, that's fine. You have variables, tick, you have equations. Well, I write something simple here, but you could also write equations involving phase variables or more complicated things, spikes, whatever have you. And then you can indeed derive this macroscopic theory, and for recurrent networks, it would be in the language of overlaps between stored information and the neuronal state. It would be correlation functions and things like that. And this was done from the 1980s onwards. But for immune networks, we don't have this yet. Well, we are trying, but we are far away from it. We don't even know what equations to write down. We can guess, but it's no more than guess. And the reason is that experiments are really tricky. It's really tricky to take parts of the immune system out and study it and expect it to behave in the same way. It's really, really difficult. And we don't know yet what we will find here. What is an appropriate macroscopic theory? And statistical mechanics is a funny way of doing things. Funny in the sense that you have a couple of go, no-go points that tell you whether at all you can use it or not. And the first point is this point, that you can take in your equation the limit of having an infinitely large system mathematically. Now, if you have this, you're in business. If you can't do this, you have nothing. Yeah? And then there's a second stage where, okay, you have your macroscopic equations and you still have to solve them. This can be hard, sometimes really, really hard, but this is the major point. This is where the magic happens. This is the magic wand. If you can do this, you're in business. So neural networks. Let's look at how it was done there. And I'll give a few pictures here of some of the early people. I think this is the dominant guy, Shunichi Amari. When I was a PhD student, I thought he must be a very arrogant man. Every paper of his, the list of references is all Amari, 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 Amari. And you look at that and you think, come on, come on. But then I actually met the man. He's the opposite. He's such a nice guy, and he really did all those things before everyone else. Yeah? So you show him something and say, Sensei, I have this nice. And he'll say, oh, maybe I have a paper that you find interesting. And he would go over. And he's done the whole bloody thing already 10 years before you. But he's the opposite of arrogant. He's such a nice and gentle person. And the other one is Coolen, who's... Well, the Spanish always love his name, I know, but he's Finnish. But these guys and a couple of others, they were the first to actually start from the McCulloch-Pitts neurons, so simple binary variables, and actually say, what happens if then they operate according to this type of rule? You have some local postsynaptic potential, which is just a weighted sum of the variables of the neurons that are feeding into it, and then a threshold function. And they ask, what happens if we take Hebb's rule literally, or we modify it, where instead of having the same patterns here, you have a pattern and a next pattern in a sequence of patterns. And they showed already, by just simulations and playing a bit without proper solutions, that in this way you create attractors, yeah, this concept of attractor networks came from them already. Where if you start in some initial state here, let's say, you move towards the nearest pattern. That was one of the patterns that were being stored. And if you did this, then you store sequences of states. So the network will just run through a pre-programmed sequence of configurations. They did all those things. But in the case of Amari, I think he's always been too early. That was his problem. He was always too early. He did marvelous things. He also did information geometry way before everyone else. And he was the victim, in that sense, of his own success. The other problem is, in the case of Amari, he published in often Japanese journals, you know, and we don't all read Japanese journals. And then came Hopfield, who didn't really do much, but he got a lot of the credit that really should go back to Amari. But he did do something nice. He basically excited the physicist because he showed that if you have symmetric synapses, the equations you write down become mathematically equivalent to what you would write down for certain magnetic systems. And he emphasized that there are interesting things that you can quantify, such as the memory capacity of your recurrent network, which is the number of patterns you can maximally store divided by the number of neurons. So, and he published not in a Japanese journal, but he published in PNAS. So, and all these physicists read PNAS. They became interested. And in those days, there were recent developments in the modeling of heterogeneous systems. And that's what we have here, the ultimate heterogeneous systems. So then came this trio of people. They're not all alive at the moment anymore. But they did a very groundbreaking paper. They took the model of Amari, added noise in, solved it analytically using the prescriptions of statistical mechanics. And they derived what is called this phase diagram, where you have here the fraction of patterns stored divided by the number of neurons, and here the noise level. And you have distinct stages where if the noise level is too high, the thing won't work, and at some point it will work as an attractor neural network, and they got this number explained, this 0.14. And some other work where people played more with topologies, they diluted synapses, and from then onwards there were a couple of waves, and I was involved in some of these. The next wave was to study the dynamics, basically. And that could all be done initially away from saturation, so with small numbers of patterns, later also with large numbers of patterns. But it all started with Amari in the early days. So physicists were really happy enjoying themselves with these models. And then many books were written. When you look at books in which these things were discussed, they were written around 1995-ish, 1990, 1995. So you don't find anywhere a good collection of results obtained from 2000 onwards. But a lot of work was done after that, and it was mainly focusing on what if we give up this unrealistic assumption that every neuron speaks to every other neuron, if we start having topology in there. And until 2000, there was no mathematical technology to look at these so-called finitely connected systems. So if you put them on a graph and you no longer say that every neuron talks to every other neuron, you have to specify the connectivity. But now we have the tools to do that, and one of the things studied was the diluted Hopfield model, and many other things could be done. The present borderline of what we can do is processes on topologies with many short loops. If you just generate random networks with certain degree distributions, you will not find many short loops. But we know in biology, in the usual systems, not just neural, but also in protein networks, you have many short loops. And that's where we don't have mathematical tools yet. So this is, at the moment, taking a lot of attention. But now back to immune networks. But because since we can now deal with finitely connected graphs, and we couldn't until about 2000, we can now go back to immune networks because they are finitely connected. Yeah? So this is where my Roman friends come in. Rome, beautiful city, one of my favorite places, after Barcelona, of course. And the key people who made interesting progress here were Elena Agliari and Adriano Barra. So these are the two on the left here, Elena, Adriano. Both worked at La Sapienza at the time. Adriano has now left. He's professor somewhere in a university slightly south of Napoli. I forgot the name, but Anna might know. And they both built on a groundbreaking paper, which was not read by many people at the time, of their supervisor at the time, which is Giorgio Parisi. He's another polymath who works on everything, right? If you talk to someone, just pick at random any problem in science, you ask, okay, what's the mathematical thing? I bet you that Giorgio has been involved somewhere. He's interested in everything. He's an extremely clever guy. But his paper was written at the time when we didn't even know the hardware yet. There was no knowledge of inhibitory T cells. So he couldn't have done what these two guys can do now. And what they said is, well, let's go and ask ourselves, what is the simplest problem that we don't know how to solve, which is always a very good recipe in science. That forget for now about subtypes of B and T cells, forget about hypersomatic mutation, forget about antigen evolving in time, just focus for now on BT interaction and find the simplest possible model that we can actually solve that describes many interacting clones. And remember, and they were aware of neural network theory because at La Sapienza there was also a lot of work, and there continues to be a lot of work on neural networks. So this is their model. And this was a really, really interesting model because you could solve it, and we learned an awful lot. And they had some very clever ideas. So we start with B clones in red, call them B mu, and each B clone recognizes a specific antigen A mu. Then we have T clones. They use the notation sigma for those. And they can coordinate the B clones via cytokine signals. And they simplified this to variables that have just 3 values. -1 if this guy, T clone I, inhibits B clone mu, 0 if they don't talk to each other, 1 if it's an excitation effect. Very simple, very clear. And then they said, well, yeah, we don't know exactly what the equations are for the B clones, but we can make an educated guess. And they said, look, we know what the main effects are. First of all, the B clone will grow if it sees antigens, so there must be something like that here that leads to an increase of the B clone. Another impact is that if we have a T clone that has a cytokine that is positive, then the product of these two guys will be positive and it will increase B. And the way they defined it, if you have a minus sign here, then if the T clone inhibits the B, it will also work out because if the Xi is negative, it will reduce the B clone. And then you add it all up over all the possible T's. Then you have captured the interaction, the antigen. Then you don't want these things to explode because there are all kinds of limitations on the hardware. So you have a decay term and you add noise. Simplest thing. Yeah? You can't make it simpler than that without doing something that is pointless. The T clones, no one knows. Yeah? That was a problem. I'll come back to that because they had a very clever idea to deal with that. Then they had to decide what to do with these cytokine variables, and they chose them all at random. They said, look, with probability C over 2n, they are actually non-zero, and then they could be equally likely +1 or -1, and then with probability 1 minus C over n, they're 0. This, in effect, means that every B clone listens to this small number C of T clones on average. So that is a small number C, and n is going to be large. So you can call that the promiscuity of the Bs. Yeah? They have more partners than one, these T partners. And you can make a picture of that. So you have your Bs, they interact with Ts, but some Ts can talk to more than one B. Yeah? And red means that it is an excitatory, green excitatory, red inhibitory. And here you see already something that has been removed in future models. Some Ts can act as an inhibitor for one and an excitator for another. But this is a minor thing that can be changed. So we should think about large systems, and there are more managers than actually people doing the work. It's just like the real world. Now comes their idea for the T clones. That was the brilliant move, I think. It was really a nice idea. They said, look, we have an equation for the Bs, it's this. But if you look at it, you can see that this you can interpret as a gradient descent on something, apart from the noise. If you take the derivative of this E that you specify here with respect to the Bs, you get that equation. So what the Bs are doing, apart from noise, they are minimizing this. Yeah? And now they said, okay, now we have to write down an equation for the Ts. If the Ts go in an opposite direction, it's just a waste of biological resources. So it is natural to assume that if it is advantageous for the Bs for this to be small, and you can see it has all the effects of the interactions in it, then let's assume it's also a good idea for the Ts to keep that thing small. They said, well, let's assume then that also the T dynamics is a noisy gradient descent on this same function. Then you work it out, you get an equation. You now know, if you buy the argument, and we will see later that it leads to very sensible results, you know how the T's evolve in response to the B's. And now mathematically you're in business, because if the noise that you add here is Gaussian, If both of these equations are gradient descent with noise, then you can write down the equilibrium probability distributions, yeah, in one go. And then outroll your mathematical machinery, you run the handle, and you can in principle, solve the model. Another observation, and now we go back to neural networks. So these papers of theirs are full of nice ideas. When you see the papers, you see there are many more authors, but forget about these other names. It's really those two that did it, Elena and Adriano. The rest is parasites and supervisors and so on. Okay, so the other observation they made is this. Okay, suppose we were only calculating the probability for certain T configurations and we were to forget about the B's. Then we would integrate this probability of the B variables. But remember, if you plug in that energy function, you see this is a quadratic thing that you get here. So this integral over the B's is a Gaussian integral that you can do. So you can do it. So you do it. And then you find that what comes out is, again, something that now involves an effective energy for the T clones. And this effective energy, everyone who's done neural networks in the 90s recognizes this. This is not just similar, this is exactly the Hopfield model. Exactly. The only difference is that here, these psi's, they are things with mostly zeros in them, whereas in the Hopfield model, it was always applied to patterns that are plus or minus 1 everywhere. Yeah? But this is now the same formula. This is not just, and this was, again, a big surprise, because there was the analogy. And if you think about it, this is the corresponding picture. So you've got rid of the B's, but the B's have been exchanged for an interaction between the T's. Yeah? You can see the B's as being the mediators of the way the T's interact with each other. And you get networks that look like this. Okay, so this is where I came involved, because to do the calculations, you have this finite connectivity, and this is more recent technology, and I've been working on that. So I really enjoyed myself working with these people. But let's look at this similarity. So this is where we are. Immune and neural networks, they all store and recall information, but they also use the same formula to do it. Yeah? The only difference is in what is the information that you put in. In the immune models, you have dilution in the patterns. Most of the pattern components are 0. Yeah? And what you then end up doing is you're recalling a very large number of patterns that each have a very small number of bits of information. And that's the opposite to what happened in neural networks, where you only recall one pattern at a time, but it is a pattern with n bits. Yeah? But that would be pointless for the immune system, because it would mean that you can only defend yourself against one invader at a time. So for the immune system, it's essential that it is like this. Yeah? So you have to maintain defenses for many. And this is a model. Now, this could still be completely rubbish. If this model, you solve it and you find that it doesn't behave like an immune system, then it's a joke, right? It's good fun, you've enjoyed yourself, move on. In fact, that's not what we will see. Now, the topologies, first of all, you see, just without solving it, already all kinds of interesting things. That if you mess around with some of the system parameters, so as alpha relates to the ratio of the number of B clones versus T clones, C was this promiscuity. Yeah, the number of T's that every B talks to. Now, if you increase that parameter C, you see that TT network at some point is changing nature. This is what is called a percolation transition. At some point, they start all talking to each other, which is not what you want. The other thing is what you see is many short loops and cliques, and this is very different from the types of systems that we would normally analyze. So then you analyze it. I'm not going to show you this because it's tricky. It is one of the longer papers that we did. It's about 45 pages of very complicated calculation, but the end result is relatively simple. But just what you have to do, you write this energy function, and you can write it in terms of overlap. So this is where our intuition from networks is so helpful, because we know how to start the calculation. We've done similar things before. You have your antigen, you have to compute some complicated sums here, and averages over the realization of all these cytokine parameters. But the thing you're really interested in computing is this, which tells you if you have an antigen trigger of strength psi, what is the response of the system in terms of this variable m, which tells you to what extent your T clones are lined up with the particular pattern that makes you defend yourself against that antigen. Okay, so forget about the details. I'm just wanting to show you that at the end of the day, you get a theory in which the key object is a function w, and you get an equation from which to solve that. Why it looks like that is at this moment completely irrelevant. But you have an explicit equation. You can take that limit n to infinity, this first hurdle that I showed you. And from that solution, you can compute all these other things, the response of the system to antigen perturbations, because you have formulas for that as well. And you find that there are different states and there are phase transitions that made really a lot of sense in a medical context. So you have one state where this is trivial, just a delta peak at 0. And the meaning of this distribution, it is the distribution of clonal crosstalk. It is the distribution of signal strength that I get from clones that are not the clone that I am myself as a T cell. So this is the perfect state where the different strategies can be used independently of each other. They don't interfere with each other. But you also have alternative solutions. And you see some funny things. In the absence of antigen, you don't see that these overlaps all become 0, and where the system starts to oscillate randomly between activated and inactivated state. This wasn't put into the model, but it came out. And this is, again, biologically vital, because we know that cells that are never activated, they just die. Or cells that are activated all the time, they also die. So this is like a homeostasis effect, where the system automatically makes sure that even in the absence of antigen, we activate all these strategies now and then. Activate them briefly and switch them off again. This comes out of the model. And you find a phase diagram in terms of, let's say, this promiscuity parameter on this axis, the noise level on that axis, where you have one phase where the whole system works like clockwork. Everything is fine, you have no clonal crosstalk. Then you cross a line and you get into a phase where you get autoimmune disease. You get clonal crosstalk. Let's say a flu virus comes in and all of a sudden you get reactions from diseases that weren't relevant here. Now, all these results can be validated in simulations. Do you know how long I still have, Paul?
How much would you need, you think, to get to your conclusion?
10, 15, 10.
That's good.
Okay. Because they don't need to show everything. So you can quantify everything. And the main thing is that you still see in the regime where everything works well, these distributions, if it's a distribution of response for clones of a given size, so it is a distribution that becomes peaked, which tells you that you keep oscillating, even in the absence of antigen. And if you do the simulations, it looks a bit like this. So you have two metastable states. You have a state where the B cell is activated, and everyone is happy. The T's are telling the B's exactly to do that, because this is activated, it activates inhibiting, but this is a negative interaction. And you have a state where the system is switched off. And in the absence of antigen, you get this type of metastability. You switch it on, you keep it on, switch it off. And this is very important. Now, further developments, of course, this is not a perfect model. It's a great model. But now you have this framework, you can make models more realistic. They took various shortcuts, which we can now start to remove one by one. There was no biological motivation for the T-clone equation, but now we have an equation. We can, in a more intelligent way, find out whether something like that might actually be happening. They had B clones that were real numbers, and for simplicity, they used binary variables for the T's, but these are minor things. We can all take them away. That identical noise levels for B and T clones, that doesn't need to be the case. Dynamical analysis. And at the level of biological detail, there are many things that we can remove, and a lot of work is presently going on to do that. This is a bit more hard and interesting also, and that is to look at the timescales where you can actually get hypersomatic mutation, which is like learning and operation at the same time, and to include antigen dynamics. And in more recent studies, some of these things were tackled. So this is a paper in which they included these idiotypic interactions of germ. Well, in a nutshell, the result is don't do this. It increases the danger of autoimmune disease. Dynamical studies have now been done in which you can see the evolution in time of these overlaps between strategies and T configurations, alternative topologies for the interaction. And this is a nice one where also various more realistic ingredients were put in. For instance, it is a more realistic equation to say that the antigen concentration and the effect of the T clones must be multiplicative, because if there is no antigen, the T won't do anything, because the T won't have an antigen either to dock to the Bs. So you can make all these things a bit more realistic. And you get all kinds of new phenomenology that, again, looks realistic in terms of what we know about immune pathologies. For instance, you see here that if you also include the distinction between helpers and activators, that you have at some stage, again, a bimodal effect where you can have a high fraction of helpers and a low fraction of regulators, or the other way around. So one of the states represents tolerance, where the system says, look, we shouldn't kick this off because it is likely to be self, and the other represents an active state. And all these things make perfect sense. Okay, I don't know whether it was 10 minutes, but I arrive at the discussion, conclusions. So the first is it was known for a long time that there is a similarity between immune and neural networks in the sense of just global features, like what are they roughly like? Large numbers, plastic, and so on. But we've now seen that it goes beyond that. Yeah? In this model of Adriano and Elena, you just get exactly the same equations. And that came out. It wasn't put in. And these models that are now solvable because we have new technology to solve them, they show a lot of rich phenomenology. You get transitions between states that make sense. You get this homeostatic switching on and off. You find low tolerance state. You can get the equivalent of autoimmunity as a result of percolation. And now I was thinking, this field benefited a lot from neural networks. Could there now be something that neural networks can learn from what we learned in immunology? And I tried to list a couple. So the first is the transfer of mathematical methods that have now been proven to work for these finitely connected systems in immunology. We can now look at models in which we take into account topological features of the network of neurons that we specify. And that couldn't be done before 2000. There was no way of doing it. Now we can. So we can look at things like degree distribution, degree correlations, modularity, all kinds of stuff like that. We can start looking at the impact of short loops, and you can do it not just for frequency-based variables, but also spike trains, phases. We could start looking at models in which the operation happens at the same time as the learning over a larger time scales. Or maybe think in terms of these functional connectivity graphs, which are also finitely connected, right? It's not everything talks to everything else and weighted, but that's not a problem. And in terms of functionality, well, these immunological systems, they were built in a way that is very similar to the neural ones, but the topological features are very different, and you can control that. And what you see in these systems is that you can recall simultaneously large numbers of patterns with each activating certain subgroups of neurons, finite numbers of subgroups of neurons. And you can get these oscillations between metastable states. So you can ask yourself, is something perhaps also happening like this in neuroscience? No? Is there something like memory homeostasis, where you need to activate now and then certain memories and switch it off again, like in sleep or whatever, to keep them alive? I don't know. So this is all speculation. Okay, thanking a few people that I've been working with, funding. If you want to read more, there's my website with all my seminars over the last 20 years or so. Coordinates. There's this beautiful building, which is, of course, is not where we are. We are in a crappy building behind that. And some selected references. I just put a small number on, because then from these references you can find your way to everything else. So these are some reviews I wrote on neural network modeling prior to 2000. And Amari wrote a recent paper in neural networks. He called it 50 years of thinking about the brain or something, because he's actually been doing that for 50 years. It's nice, short paper, non-technical paper. Then immunological work prior to 2000, where George is really the odd one out, because this is really very different from these. But this was the motivation for the other work by his PhD students, who are now developing this. And there are some of the references to that here. Okay, thank you very much for your attention.
Thank you. Fantastic talk. All right, any questions? Tony.
Thank you for a really fascinating talk. I'm very ignorant about immunology, so but I imagine that T cells and B cells move around a lot, which neurons obviously don't. So do you take into account that? Are you thinking about that? And generally, are there spatial effects that we want to think about?
Yeah, So remember that the variables here are not the cells themselves, but the concentrations of cells. So it's assumed already that they're spread out. But there are spatial effects in the sense that certain types of B cells will only be found in certain organs, or at certain stages they will be found in certain parts of certain organs. And this, of course, is part of this next stage of making things more realistic. And once you start thinking about subtypes of B cells and T cells, then immediately you have to think about what you just said, because they will only be able to interact with others that are in the same location, right? And then, so this will become relevant in the next wave. Yes.
So Ton, you also mentioned that, okay, there was this older model about memory in the immune system that would say it's like this flip-flop idea, right? You have sort of the antigens and anti-antigens, and they sort of cyclically reactivate. And then in this reverberation, you have your memory. This was now thrown out of the window.
Yeah.
But now, in your model that you present here, it doesn't give you an alternative view on then what the memory substrate would be in the immune system, because that's the fundamental problem we got to get a handle on.
Yeah, I mean, if you were to talk to a biologist here, he would say, no, we understand it. It's just these long-lived memory cells. There is no problem. Yeah, and who am I to say that they are wrong? But to me, it's still not those. It just feels to me like a cheap way out. But maybe nature does it that way. But it feels to me like a bit of a cheap way out. I would like to see stronger evidence for there being actually cells. Let's say a B cell has been in action. It has found a way to protect you from some disease. And then it all of a sudden transforms into a cell, and it hides somewhere. It stays there, and it lives for 30 years. I don't buy that. It just doesn't feel right to me. But I can't present an alternative, so I have to shut up and say, okay, let's go with it for now.
So the model actually would have that responsibility to give us an alternative, no? Don't you think so? And the second thing is that for a model, models are not right or wrong, right? They're just a way of thinking about the phenomenon.
Yep.
And then the validity you would find in their empirical support,
Yep.
right? Or their consistency with the data. So have you been able to already generate predictions with the model that have been tested in an experimental setting?
Yeah. So there's one project that I'm doing with one of my PhD students, and we're involved in a partnership with immunologists and bioinformaticians. And one of the things we there try to do is really see whether we can derive equations like this, or maybe others, starting really from the biological hardware, doing the hard graft of going to all these biological details, reading all these papers, talking to the immunologist, and trying to derive it from scratch, because this is exactly where I want to go. But it's very difficult to talk to biologists in general, maybe, because they don't like reducing models, you know? To them, the best model is the model that has all the ingredients that have ever been measured in experiments. And I always keep telling them, I don't want to build the Maserati yet. I want the Volkswagen. Give me a list of the ingredients and rank them for me. Say, the first thing, if this is not in your model, I'm not even talking to you. The second thing, I might talk to you, but I think it's pointless what you do. The third is, maybe we can have a coffee and it becomes interesting. So rank for me these things. And it is really very difficult to get them to do that. But we try. And we are now educating each other. Let's say they teach me a lot about the biology, we teach them a bit about the modeling, and the bioinformatician teaches all about this recombination, the structural features of the receptors and all that. So we want to go there, but it is a road that will take time.
On the other hand, they might say it's really impossible to talk to these theoretical physicists, right?
I mean, all disciplines I've learned have their own foibles, and physicists
no,
can be arrogant beasts, and what they also often don't want to do is to get their hands dirty looking at real data.
Sure.
That's the
No,
we all have
they
our...
present you with a spherical cow, right?
Yeah. If not a point cow.
Yeah. Exactly.
Yeah.
But now the other thing is that you could also argue that either you, with your friends in Rome, you stumbled on something extremely profound, or
Well, they stumbled onto it. I was helping.
you are their ambassador in this audience,
Yeah.
or maybe you found something that's really trivial. Because you can say, look, we found a characterization of the macroscopic dynamics of an immune system that's compatible what we found for the neural systems with the Hopfield network. We can think about it as operating in a macroscopic energy landscape in which I'm sort of performing gradient descent to find my solutions, right? So the solution space is now defined, and I know how to control that. But if you now look at the impact that this way of thinking has had in neuroscience, then actually, in my opinion, the mileage has not been so great. Yes, it's used a lot, okay? And then people talk about, okay, memories are attractors, and we can characterize it. And then Sompolinski and Amit and all these guys could write many papers about that. But if you then pose the question, what has been really the empirical impact in the way people did their experiments and really sort of extracted information from the system? We'll look at what we looked at this morning, right? How did it influence the way we try to manipulate brains that don't really operate anymore in a sort of a normal way? Then actually, the impact has not been that huge. Okay, of course, many people disagree with me, particularly Gustavo, who was sitting here earlier. But in my opinion, that empirical impact, to say it has really advanced our fundamental understanding, has not been so massive, because the problem is you are still arbitrarily defining your state space, right? You are saying, well, if you talk about a memory is some a stable piece of information that you can carry over time. And defining it in that way, it gives you a state space that has the features that satisfy your formalism, right? There's a circularity there. So is it trivial, or is it really profound what we're looking at here?
It's a philosophical question, Paul, and none of these models, as you said already, all models, you phrased it like some models are right, some wrong. Also, one famous person said all models are wrong, by definition, because there's always the next thing. If models help you to understand certain phenomena, that's all they're meant to be doing. And if you learn and then you move on, fine. And if it turns out that later other aspects of a system turn out to be more important, either because of your practical desire in medicine to manipulate them or whatever, then there's no issue. The main thing I think you should avoid is thinking, what some people sometimes try to do is that they come in with an idea and then they don't realize that it's just one idea, but everything else that was done until now was rubbish, and this is how it is. They're so naive, right? And that's true for attractor networks also. It's true for this model also. It's not how science works. We build on each other's ideas. And this arrogance of thinking that I now know that everything that everyone else said in the past was rubbish, and this is not a big thing, and I give it a name, I invent a fancy name. No, we know that this happens, but it is not helpful. So attractors, yes, I mean, they've been useful in helping us to think, but this whole idea of fixed point attractors is complete rubbish for neural networks in the first place because of Dale's law. There are no symmetric synapses, so there is never an energy function on which you walk. But it was a helpful concept. And now you can start thinking about these pictures of Escher, where you keep going round, because that's more like what you have if you don't have symmetric synapses. Here also, this will go. We know it will go. And the sooner it goes, the better, because it will go for a very good reason. Namely, we bring in antigen dynamics. The antigens aren't static. It's a rat race. They grow, they expand, and you hope the immune system kills them before they actually grow. Immediately, all the equilibrium pictures go. It doesn't mean that you haven't learned a lot, right? It doesn't mean that you can't build on that and you take the next step. So I don't have a problem with this. Science is a whole process of ideas being replaced by better ideas. They have their lifetime.
Sure. Okay. Other questions? All right. In that case, Don, thank you very much. Fantastic talk. Then a message to the young people. In the lab, we have this rule in our meetings, which is one brain, one question. Every brain in the room is supposed to pose at least one question. And that also serves a purpose, because if you have a mission in life, which is, as part of the audience, you have to pose questions, you always work on the assumption that whatever talk you hear is wrong. You just have to find where the error is, okay? So you have more acuity. So I think also for everyone here in the room, commit yourself to that, right? Really, for every talk, try to find that question that you think really can sort of disprove some of the main contentions that the speakers present to you, right? So stay awake, pose your questions, be part of the story, and show how the old guys are all wrong. Yeah? All right, so now...
Never forget the motto of the Royal Society. It's one of the greatest things. They have this motto, I don't know the Latin word for it, but it means never trust anything on authority alone.
That's right. All right, you got it. Now you can enjoy your lunch. which on better authority, I'm told, is in our lunch tunnel.